So, and from that, you started to get changes in the development of the creatures so that they would have a spinal cord, which then sent out branches to each segment. And the blood vessels also would send out arteries and veins coming back at each level of the segments. And after that develops, you've got things like sharks with cartilage in their back. And then they get calcified, and we now are into the vertebrates. Yes?
When did the brain happen?
When did the brain happen? Define brain. Because those worms,
Yeah.
the worms, these guys, they had a nervous system with little lumps in it. Ganglia, they're called. And they're the brain of the worm. If you're talking about a brain such as ours, you really have to wait until you get, you're going from worms, then you've got mollusks and, clamshells and so on. And they have a nervous system, and clams, you may know, have eyes. They have light sensors. So if you ever see a clam open underwater and move your hand towards it slowly so it doesn't get the flop of water, move your hand so that there's a shadow over that, and it'll clamp closed.
Yeah.
Because it has tiny little light sensors, and if you've seen pictures of them, they'll be either a brilliant blue or red or something, the little tiny specks. So the Hox genes are genes that control other genes. Well, how do they do that? It's not in the handout, you'll have to write this down. This last year, scientists discovered that active genes are near the center of the nucleus. Cell nucleus, DNA. It turns out that the outside of the nucleus is a place where the DNA gets shoved, coiled on itself, and then coiled again. It's hypercoiling. And that involves certain proteins. And the proteins only attached at certain points. And obviously, if you make a lot of those proteins, a lot of stuff is going to be inactivated. Because you can't get at DNA. Where's one that's okay? You can't get at it. You can't, get at it if it's all bound up. Mm-hmm. But if it's opened out, you can get at the DNA. Sorry, Nico, please. Uh-oh. Well, here, Rex. Try and catch that one.
Thanks for drawing my picture.
Pretty good, huh?
Yeah.
The idea is that, well, wait a minute, I need one.
Here.
The idea is that when it's together, the DNA cannot be read. It cannot be transcribed. The genes are inactivated. They have to be opened up. And that happens in the middle. And we only just now found that out this last year. That one of the controls is not simply the manufacturer of a protein in a feedback system that gloms onto the area of, turn on or turn off to prevent it from turning on, the gene. That's one mechanism, and we've been aware of that for a long time. But this is on, the next level of inactivation of DNA. So I should have drawn these in the periphery, like this. Because those are completely quiet, and the ones in the middle are active.
To use your term, are you saying those are folded, but then they unfold within the nucleus also?
Yeah, if this section of DNA needs to be activated, it will get unhooked from the wall, and it will uncoil. So now, this part of the DNA, which used to be inactive here, can become active
And readable.
and readable, so that the genes can actually make the proteins that do the things that happen in development.
What initiates the change?
We have no clue.
It's a-
No, that's one of the things, actually, if you think about it. The sperm is just DNA with mitochondria and a tail. There's not a whole lot there that is involved in actual embryological development. The egg is formed through an extremely specialized form of division and creation. And it's my personal opinion that it's the egg that has all of the things that set into motion what happens when. That's my own personal opinion, so don't take it with a grain of salt. Okay, I wanted to bring up a phrase that you may have heard. There is a phrase from what's called the recapitulation theory, which states ontology recapitulates phylogeny. Ontology recapitulates phylogeny. What does that mean? In the 1866, one German zoologist said, you know, if you look at those embryos that I showed you, the ones that all look so similar, where are they? Here we go. His thinking, and this goes along with the arches, the gill arches, his thinking was the embryo actually evolves. It recreates the evolution of its kind from one cell. You know, everything that we are talking about starts as one cell. So it seems kind of logical that fossils started with one cell and became multicellular. Embryos start as one cell and become multicellular. So he said, I think that development of the embryo, which is the phylogeny, no, the ontology, and phylogeny is the evolutionary history, the path that it takes. This has been disproven, and I only mention it because if you hear ontology recapitulates phylogeny, you'll know that that is in fact not quite correct. However, the statement that related animals have very similar embryologic formations is true. And I gave you some numerous examples in here so that I wouldn't have to go over it. I mentioned previously Tiktaalik, which was the Tiktaalik was the fish-like animal. Here we go. Okay, this shows. Remember we talked about missing links, or I may have? There is no such thing as a missing link. Everything is evolving at the time it is living. Evolution doesn't stop. You don't end up with an end form. You don't have anything in between. We are missing links. 100,000 years from now, if somebody dug up my fossil, if I fossilized, I've been thinking of asking somebody to do that. Well, it's just for the hell of it, to see if they can do it. No, immortality in some way.